This grand round is part of the CTSA Visiting Scholars Program. The Scholars Program is administered by CCOS, the Coordination, Communication, and Operations Support Center for NCATS. Our CTSI partnered with 42 other CTSA hubs to provide an opportunity to KL2 scholars to make connections that will remain beyond this visit. As a host site for the Visiting Scholar Program, we're happy to have been matched with a scholar from NYU, Dr. Giulio Qaurta, our presenter today. Dr. Qaurta is an assistant professor in the Division of Gastroenterology and the Department of Medicine at NYU. As a practicing gastroenterologist, he observes firsthand the impact of the gut microbiome on the well-being of patients, as well as the shortcomings of existing antimicrobial therapies. The concept that gut microbiomes have an effect on our health is novel, though it is now recognized that numerous chronic diseases, including obesity, chronic liver disease, and heart disease, are linked to the physiology of gut microbes. Since his undergraduate studies, he has been involved in studying non-coding RNAs in mammalian and bacterial genomes in the laboratory of Tamar Schlick. His research at that time was focused on understanding alternative folding of RNA and interactions of bacterial regulatory elements, riboswitches, with small natural metabolites. These interests eventually led to a research fellowship in the laboratory of Martin Blaser during medical school, which was focused on understanding mutational capacity of H. pylori and searching for new RNA elements that control gene expression in bacteria. This study further solidified any interest in developing therapeutics aimed at RNA molecules. The title of Dr. Qaurta's talk today is Exploring Metabolic Pathways in Gut Microbiome Through RNA Riboswitches. And with that, I'll hand it over to Dr. Qaurta.
All right, thank you for that wonderful introduction. And I'll just make sure, can everyone see the slides okay? All right, great.
Yes.
All right, so yes, thank you, Jessica. My name is Giulio. I'm one of the physicians in the Department of Medicine doing research at NYU. And I'd be happy to present my work on riboswitch distribution in the human gut microbiome, revealing common metabolite pathways. All right, so I'll start today's talk by introducing the idea of the gut microbiome and what it is. I'll talk a little bit about riboswitches and what that class of RNAs encompasses. I'll talk about our recent work on the distribution of riboswitches in the gut and variation in riboswitch distribution on an individual level. So the gut microbiome encompasses all the bacteria, yeast, fungi, viruses that inhabit the intestines of the human body. And the gut microbiome is particularly interesting because it houses the greatest diversity and the greatest number of bacteria and microbes than any other site in the human body. In recent years, the gut microbiome has been linked to a number of different diseases. And the more and more we look at the gut microbiome, the more we find that it is being linked to a number of different metabolic processes. Diseases as diverse as neuronal diseases, neurological diseases, metabolism and nutrition, and even cancer. I would argue that the way that all of these different diseases are linked to the gut microbiome is through the secretion of metabolites. One of the major ways microbes affect human physiology is through the exchange of metabolites. And what I mean by that is that these are chemicals, some of them may be beneficial, some of them may be harmful, that are exchanged between the host, the diet, and bacteria. And some of the questions we can ask are what the metabolites that are secreted by the gut bacteria, how they affect host physiology, and in what conditions are they made. And here we have an example of some of the different bacterial metabolites which are known to be made by the gut. So the overall concept and the overall idea is that dietary precursors and host-derived molecules can be used by bacteria that live in the intestines, and they'll then convert that into an assortment of different chemicals that then have an effect on the human host. For example, certain peptides and certain polyketides can be made through non-ribosomal synthesis. Short peptides, post-translationally modified, can enter and affect the host physiology, affect the host cells, and even amino acid metabolites can be taken up directly into the host circulation. As a physician and a scientist, I often ask, how does this happen? And framed in a different way, you can also ask the question, how is this process regulated? Okay. And here's where I'll introduce the idea of riboswitch. And this is a class of messenger RNAs that this is a class of non-protein coding RNAs that are part of the messenger RNAs of an assortment of different bacteria. And here what we're looking at is a riboswitch that responds and binds to a particular flavin called FMN, flavin mononucleotide. And you'll notice that this particular RNA, it's about 250 nucleotides, has an upstream part, which we call aptamer. And in that part, that's a sensor domain for a small molecule, FMN, or metabolite. And on the other side of the coin, we have downstream a gene expression platform, um, a gene regulatory element that regulates the expression of the gene that's linked to this particular riboswitch. So let me go into this in a little bit more detail. There are over 45 classes of riboswitches that are known to be present in bacteria with metabolites that bind to riboswitches as diverse as heavy metals, as diverse as small molecules like second messengers, amino acids, and even B vitamins like thiamine and cobalamin. And the property that links them all together is that they bind to these different metabolites that exist in bacteria with very, very high affinity. So they tend to have dissociation constants in the nanomolar range. And the other important thing is that uh, in general, one riboswitch will bind to one particular metabolite. So it's a one-to-one type of interaction. I've had the real pleasure of working in the laboratory of Dr. Evgeny Noodler over the past 5 years, and he's credited with some of the first descriptions of riboswitches in E. coli and B. subtilis. And his group helped show that these riboswitches are regulating the expression of genes in bacteria in response to very specific metabolites or small molecules. Okay, and let me show you how that works in a step-by-step process. Okay, so how does a riboswitch work? In the first step, it all starts with the process of transcription. Okay, transcription is the way that humans and bacteria and fungi make ribonucleic acid or RNA. So the RNA polymerase enzyme will bind to a particular region of the genome, and it'll start making mRNA, messenger RNA, from the gene sequence. Okay. And the riboswitch is part of that messenger RNA. It tends to lie in the very early or start portion of the RNA molecule. In the absence of its metabolite or absence of its ligand, the RNA will fold into a very particular structure during transcription, and this is really important. In this case, what we're seeing is that an anti-termination structure or fold will be favored in the absence of its ligand. That anti-termination structure permits the translation of a protein to which that messenger RNA is attached. So translation is the way that a ribosome will make a protein from that RNA, okay? Then the expression of that protein will enable the transport of a very specific metabolite. So in this case here, in the example in the cartoon that I'm showing, this particular gene, it encodes for a protein that enables the transport of a very specific metabolite or ligand from the extracellular environment of the bacteria to the intracellular environment, okay? That metabolite will then get taken up, and in the next round of transcription, that ligand will have a direct interaction with that aptamer domain that I mentioned earlier, and that will cause a formation of a different structure in the same RNA sequence that then stops the RNA polymerase from proceeding down the length of the gene in a process which we call termination. Once the termination step happens, that RNA is then degraded, and that feedback loop is now in balance, right? The amount of ligand in the cell is balanced with the amount of the riboswitch activity that we started off with. So the important point is that I hope that everybody can kind of appreciate that metabolite levels are somehow genetically encoded by riboswitches and their conformations, okay? Taking another, looking at it from a different perspective is we can ask, how is riboswitch mediated regulation studied in a model system? And so here what I'm presenting is a riboswitch that's exists in the E. coli genome. E. coli is a normal inhabitant of the gut of humans and mammals. And inside of its genome, we have an area that encodes for a riboswitch that regulates the expression of B12, vitamin B12, which is otherwise known as cobalamin, okay? So BTUB is a vitamin B12 transporter. That's the role of that particular protein or gene. And if the B12 level in the cell is low, this particular riboswitch will allow the expression of BTUB protein, and B12 is imported into the cell. What happens when a lot of B12 starts coming into the cell is that that particular switch will change structure, kind of like what we saw before, and the expression of that particular gene or protein will stop. And BTUB is no longer going to be expressed, and the B12 level is going to be kept in check, okay? So this is a way that bacteria interact with their environment to kind of balance the way that these metabolites, B12 or FMN or thiamine, how they strike a balance between their environment and their intracellular metabolic capacity, okay? The way we work with these particular riboswitches in the lab is we might work with them in a plasmid. And what we might do is we'll fuse this BTUB gene, or at least part of it, with a fluorescent protein called GFP. And that'll give us sort of a readout of the B12 capacity in the cell and outside. And what we might usually see is that the expression level of BTUB will be correlated with the B12 in the cell and in the media. So when we spike in B12 into the media, the expression of BTUB will be low. And in the opposite case, B12 low in the cell, BTUB expression will be high. And we would sort of decode this or kind of study this by actually looking at how the cells express this GFP, this fluorescent protein. Let me show you an example of that. So looking at it in sort of another kind of cartoon way, on the left, we're seeing the case where B12 is spiked into the media, into the cells, and and in when the B12 in the cell goes up, GFP will drop. And what we see on the bottom is an example where the E. coli cells they're not so bright. They're not expressing that particular GFP protein. And in the opposite case, when we don't add any B12 to the media, the GFP expressed by these E. coli will actually be much higher, in concordance with the BTUB level, kind of. And the E. coli cells will become bright green because they are expressing this very fluorescent protein, GFP. All I did, or as a gastroenterologist, I started to think about this in sort of a broader context. Like, if that's what's happening in the lab, what about what's happening with these riboswitches in the lumen of the intestine? Maybe this can give us sort of a clue or an idea of what's happening with the metabolite balance that's happening in the intestines. Okay. So the first question we asked was, can riboswitches teach us about the balance of metabolites in the gut? And I teamed up with my prior mentor, Tamar Schlick, to ask the question, how do riboswitches distribute in the gut microbiome? And what we used was one of the biggest collections of non-redundant genomes from gut microbiome samples from around the world. So this was an international collection of over 204,000 genomes of bacteria coming from human samples. And I used the representative set, which was over 4,700 genomes, and I cross-referenced that with all the known riboswitch classes that are to date known in a database called RFAM, the RNA Families Database. We used a tool called Infernal to search this data set for the riboswitches, and we found over 36,000 high confidence hits across the entire span of the gut microbiome. At the same time, we also mapped the protein coding sequence, and we found over 10 million proteins which the gut microbiome codes for. And that amounts, if you could do the math, about 0.3% of proteins are regulated by riboswitches, which sounds like a very small number, but I'll just kind of mention here that riboswitches don't just control one protein, but they regulate the expression of entire operons, which can include 4, 5, or even 6 different proteins encoded on one messenger RNA transcript. So the amount of regulation which these riboswitches entail is still kind of an open question for us. And so here's what we found. What we're looking at here is the distribution of riboswitches in the gut microbiome uh, in sort of a proportional level. So the amount of each riboswitch class is plotted by color. And so you could see that number 1 here represents the thiamine pyrophosphate riboswitch class, and number 2 is the cobalamin riboswitch class. And out of those 40, in reality, 45 different riboswitch classes, that amounted to only 22 that were present in the human gut, which was really interesting to us because it means that not all riboswitches are found in gut bacteria, but rather um, a very select subset. Okay, so these were the 22 different riboswitch classes we found. And when we broke it down in terms of the class of metabolites, you can see that the majority of the riboswitches in our human gut are devoted to the production and the balance, really, of B vitamins. So things like vitamin B1, B12, which is cobalamin, and B3 and B2 are devoted riboswitch classes in the gut bacteria. Another major class are what we term essential metabolites. Things like S-adenosylmethionine, which is the major methyl donor within biochemistry, and other things like amino acids like lysine and glycine, purine metabolites. And finally, the third major class, which the riboswitches are responsible for controlling in the human gut, are devoted to toxins. So things like guanidine and manganese and homocysteine, those were minor classes in the human gut, but nevertheless, they were there. And we'll come back to that in future slides. When we looked at the human gut and compared that against the mouse, the oral, and the marine microbiomes, we found that there was sort of a distinct proportion or distinct balance between the riboswitches in all of the gut bacteria. So, for example, the marine microbiome, you could see that pink box at the bottom. That's the glutamine riboswitch, and that was present only in the marine environment, as opposed to that being completely absent in the human gut. So that told us something, that maybe the abundance of these different riboswitches is actually evolutionarily conserved and important. We also looked at the distribution of riboswitches across bacteria. What we found was that if you look at the x-axis, each box represents a different riboswitch class, and the y-axis that is looking at a different bacterial phylum. And phylum just is a fancy way of saying a different bacterial class. So when you look at the bacterial classes that have riboswitches and those riboswitches that are sort of represented in many bacterial phyla, we see that the thiamine pyrophosphate, the vitamin B1 riboswitch, is not just present in high abundance, but it's extremely diverse. So it goes along many, many different bacteria, is suggesting to us the importance of B1 balance in the human gut, okay? The same was true with cobalamin and flavin mononucleotide. But the other thing to kind of keep in mind is that there are some riboswitch classes like molybdenum or cyclic diadenosine monophosphate that are present in very select few bacteria. And so the idea of dysbiosis is that we might lose, maybe after a course of antibiotics or after exposure to a toxin in our environment, we might lose some of the important bacteria that are normally there in a healthy gut. And along with that, we might lose the ability to metabolize some of these important intermediates in central metabolism. The other really interesting thing about this data set is that we could parse it by geographic location. So we knew from the data set where the samples were coming from across the globe. And it's been known for many, many years that everybody's gut microbiome is a little bit different. It depends not just on our environment and our diet, but it also depends on our genes. So there's this sort of a important balance that the gut microbes will have with the host. So, for example, the North American gut microbiome is different than what someone in Asia might have in their intestines based on the taxonomic representation or the classes of bacteria that are there. Okay, you can see that the bars, the red bar, the firmicutes, the absolute amount of firmicutes in North America versus Asia is slightly different, and fusobacteria are almost absent in North America, but are kind of enriched in Asia. But when we look at the different riboswitches that are there, at sort of the different metabolic regulators, if you will, what we see is that across Africa, Asia, Europe, North America, Oceania, South America, that there's a relative preservation of the balance of these different riboswitches. So it tells us that possibly there's evolutionary pressure to conserve the balance of important metabolites by using or exchanging different bacteria from the environment, which is really interesting. So, in other words, the lack of the presence of fusobacteria in Asia might be used might be selected for because the fusobacteria actually provide an important ecological niche that the fusobacteria in North America don't. So that's sort of the idea that the metabolome or the all the metabolites that are in the gut are somehow evolutionary conserved. Okay. All right. And then we also looked at the average number of riboswitches per genome, and we found that there was a huge diversity in terms of the number or the distribution of riboswitches in each bacterial genome. So fusobacteria tend to have the most number of switches, and Myxocoda tend to have very few. But in firmicutes, there's a huge range, with some of the highest being expressed in a bacteria called Peribacillus simplex, which has over 60 examples of riboswitches controlling and responding to different metabolites in its genome. What we also did was we looked at the different pathways which these riboswitches are associated with. And as we kind of mentioned before, riboswitches are mostly associated with the feedback regulation um, of the metabolite in the cell. So in this case, we're looking at the cobalamin or B12 riboswitch distribution across the gut. We see that it's highly enriched in genes that are part of cobalamin synthesis, biosynthesis. So the amount that cobalamin is made de novo in each cell. But you can also see that's really just the tip of the iceberg, that cobalamin riboswitches uh, are associated with pathways that go well beyond just cobalamin biosynthesis. So it speaks to the importance of a non-canonical role for riboswitches as well. All right, so I'll just pause here and I'll say, in summary, there are 22 classes of riboswitches found in the human gut microbiome. Most are controlling and balancing the levels of B vitamins in the gut. The abundance of riboswitches in the gut microbiome is relatively conserved across continents, and we brought up the question of a possible evolutionary conservation there. Most riboswitches are found abundantly in fusobacteria and firmicutes. And finally, riboswitches are found associated with a variety of different genes and proteins, but are mostly devoted to the transport of metabolites in and out of the cell. All right, before I go on to the next section, are there any questions? All right. So the next thing we asked, how does this sort of global analysis compare to what we might expect on a metagenomic analysis on an individual level? So in that case, I used a study that published the gut metagenome of 110 healthy individuals. These were the average age was 29 years old, and their BMI was 21. So these were individuals that were not overweight and not underweight. They had normal weight, and they had no past medical history, and there was no family history of diabetes. And the investigators uh, acquired fecal samples, they performed DNA extraction, then they did something called shotgun metagenomic sequencing, which they sequenced all of the different DNA, bacterial DNA that was there. They ended up with about 500 million paired end reads. They assembled those reads into contigs, which are contiguous regions of DNA. And then I used those contigs to scan infernal, just like I did before, for riboswitch aptamers, okay? And here was what we found. Um, here's what we're looking at is the abundance of each riboswitch class in this new data set, according to the number of in each across the entire sequence data set. So um, in the first case, we found 12.6 on average, 12.6 copies of the thymine pyrophosphate riboswitch. And then in decreasing order, we found all of the other aptamers as well. What was nice to see was that we had the same general pattern or general trend in terms of the riboswitch distribution of aptamers across the gut metagenome. But what was particularly interesting for me, actually, as a physician, was that some of the patients actually had sort of higher copies than what would be expected in these sort of metabolites that help bacteria uh, degrade toxins, things like guanidine or trimethylamines, which are present in fertilizers, um, homocysteine, which I'll go into in a little bit, and manganese, which is a heavy metal, which is toxic to both. We should only get very, very minute amounts of manganese in our diet, and too much is not a good thing. So I'll go over the case of homocysteine. When I was in medical school, we talked a lot about homocysteine as a biomarker of coronary artery disease. And homocysteine levels were noted in two landmark studies out of the New England Journal to be correlated with all-cause mortality or all-cause cardiovascular mortality. The higher the homocysteine level, the higher the mortality, or the sooner patients would die or less survival, okay? It brought to us this model or this concept that our diet and our environmental exposures um, are sort of sensed by the gut bacteria that normally reside within the intestine. And possibly one of the sources of homocysteine is actually through the conversion of dietary factors into homocysteine, which can be taken up by the liver and then in a second effect cause atherosclerosis or be related to atherosclerosis in some way. And in my mind, I thought riboswitches could sort of be a marker of the metabolic capacity of the degradation of these toxins. That was an interesting idea that came out of this study. The other important question was, could riboswitches serve as a fingerprint of metabolic balance in the host? Finally, another sort of overarching question is, why is there such a wide difference? If riboswitches are sort of a very basic and important part of bacterial physiology, why were there so few? And why is there such a big difference between the abundance of riboswitches in fusobacteria versus something like Myxocoda? And one of the reasons why is that we are focused in the lab on an idea of these orphan riboswitches. And these are switches which are predicted with high accuracy, but we don't know what they work on, and we don't know quite yet what they bind to. And you can see that here in the darker gray that they actually make up, one of the top 10 classes of riboswitches across gut metagenomes. So it's actually the importance of riboswitches and the idea that they actually have some role in the gut is an evolving concept. So here's how we kind of use computers and uh, statistics to kind of predict where riboswitches are found in bacterial genomes. And with pretty high accuracy, we can kind of see sort of the areas where riboswitches are expected, and we can see that they are predicted to fold into complex structures that are characteristic of aptamers. And so we have a pretty good idea of where they might be across the genome. And I'll talk a little bit about my work on an orphan in Helicobacter pylori. H. pylori is a bacteria that normally lives in the stomach of humans. It only lives in humans too. It evolved. You can find H. pylori going as far back as ancient ancestors in Africa. And one of the key orphans in the H. pylori genome is the HOPC riboswitch. And what's known is that mutations in the HOPC switch, in the HOPC aptamer, and the riboswitch, are associated with disease. So if you mutate it in certain areas or other areas of the riboswitch are changed, it can be associated with diseases like peptic ulcers or gastritis, even gastric cancer or lymphoma. Okay. And molecular biologists and microbiologists have also worked on characterizing what the protein associated with the HOPC gene does. What does it do? And when they knock it out, when they knock out this gene, they see that Helicobacter, uh, when it's tagged fluorescently, it adheres or sticks to the mucosa of the stomach a little bit less. And so that tells us or suggests to us that this particular switch is somehow related to the process of adherence to the host. So which is very interesting. It's part of the basic biology of Helicobacter pylori. And so what we did in the lab was we put this particular switch in uh, in our plasmid reporter that we kind of talked about earlier. And what we did was we shocked the cells with acid. We gave different amounts of acid to the E. coli cells, and we found that there was an induction in the riboswitch activity when we acid stressed the cells. So going from pH 7 to pH 3, we see that the absolute expression level of this particular gene, this riboswitch, goes up dramatically. On the right, I'm showing a control, uh, a, a different region that is predicted not to have a riboswitch at all. And you can see that between pH 7 and pH 3, there's really not much of a difference. So this acid effect seems to be specific for the H. pylori riboswitch. So what we did was we developed an assay where we can actually mix this particular riboswitch with a mixture of different compounds and try and learn what it's binding to. And I won't get too far into the details too much, but I will tell you that after a long and arduous process, we were able to uh, see that we can pull down riboswitch associated metabolites with pretty high accuracy. So this is working with our metabolomics core at NYU. And what I did was I practiced on a riboswitch aptamer that is known to bind to adenine. And when we looked at all the different metabolites that are there, we found that the adenine abundance in the pull down was one of the highest compounds there. So this was very nice to see. And then using this pull down, we can also actually do something called SHAPE, which is a structural probing. I don't want to get too in depth into it, but basically what we see is that when we use the metabolite that we pull down in a shape assay, we see that there are some important structural changes. All right, so then I just wanted to mention also in the closing some different limitations of our study. So what we use is the overall architecture or topology of these different riboswitches to search genomes. So we're using sort of this RNA families database, which basically has all of these sort of RNAs uh, coded for by secondary structure. There are certain areas in this particular RNA which is predicted to bind to S-adenosylmethionine that should be base paired. Some areas should be single-stranded. Some areas should have a G or an A. And so we use that code to scan the entire genome as input. And the idea is that when we find a hit, that this represents a bona fide riboswitch domain. And we used a very, very stringent cutoff to make sure that we were finding true riboswitch aptamers. But in recent years, it's been shown that sometimes riboswitches can have the same exact topology or the same architecture, but bind to a completely different metabolite. So here's one example that was recently published of a SAM switch, something very, very close to a SAM switch, but actually a couple of key mutations, a couple of insertions leads to the ability of this riboswitch to actually bind to spermidine, a polyamine, as opposed to S-adenosylmethionine. So although we use a very high stringency for these riboswitches, it is possible that the picture that we're getting, it could not truly represent what the riboswitches are binding to, what they're controlling in the different cells. All right, with that, I'll close, and I'm happy to share our recent publication on this work with myself and Dr. Schlick in the Journal of Physical Chemistry B. And I'll also thank my mentors of Jenny Noodler, Tamar Schlick, Sasha Serganov, Drew Jones, Avi Rasuli, our microbiologist expert in the lab, my clinical mentor, Mark Buchapin, my research mentor, Ozma Shakat, our KL2 director, Francisco Castellanos, my team members, Nikhil Shah and Jerry Zhang, and I thank my funders at the NYU School of Medicine, the NIAID, and KL2, and through the ANCATS. So thank you very much.
That was great,
Thank
Giulio.
you.
So I have a question actually, Giulio, about I don't want to take all the questions, but I have a lot. But
So
one thing that I was really curious about was the section that you talked about with the H. pylori.
yeah,
I just thought that was really interesting. And by the way, I absolutely love your fingers and your slides. They're like exemplary, you know,
oh,
not
thank
too
you.
cluttered, really nice drawings, all of the things. But you had mentioned something about the acids, you know, when you did the acid stress, you're seeing the increase in the ribosomes. And I'm just thinking about peptic ulcer disease and GERD and all the things, and just
Oh,
wondering,
yeah.
like, you know, being that we're in clinical translational science, where do you see that specific aspect of the work going? What do you think the implications are of that?
Yeah, it's really intriguing to think that, this particular bacteria, which is known to cause cancer, to cause peptic ulcers, as you mentioned, is somehow responding to and binding to the host in response to some kind of acid-related metabolite. And so, I mean, the translational potential of that could be that if it were to work, and we're still just sort of learning about this, is if it were to work, could it be possible to kind of, instead of killing and sort of making destroying all the bacteria that in response to antibiotics, because H. pylori
Mhm.
treatment is really heavy. It's
Yeah.
four antibiotics.
Hmm.
Um, and instead of killing it, if we could say, okay, actually, we just want to sort of do like a little molecular trickery and say, okay, please just fall off and just don't bind. That would be really good. And it would be sort of maybe specific to H. pylori. That's the pipe dream. That's 10 years down the
I
line.
love that. Yeah.
But I think what we can do in sort of the near term is sort of a very kind of something that we have in process, and it's actually really hard to isolate H. pylori. But
Yeah.
during endoscopies, which I do here in New York, um, we can isolate H. pylori, and because sequencing is feasible on a lower cost basis, now we can sequence the genome and we can see if this particular switch is uh, if it's conserved and if it's sort of a feature of resistance, antibiotic resistance, or is it a feature of maybe um, people who have peptic ulcer disease? Maybe it's a little bit more
Right.
active or multi-copy. So you can ask some interesting questions on the sequencing level too.
Yeah, that's great. I just thinking about, you know, and the antibiotics are pretty brutal for people. So yeah, I liked what you said about the trickery and that. Other questions? Yeah.
Well, I just wanted to say thanks very much for this talk. My name's Lauren. I'm a KL2 scholar at the Hershey, the Penn State College of Medicine. I have no intelligible questions about your content because I'm a psychologist and, we kind of swim in different waters. But I, like LJ, really liked your figures and your visualizations. So I'm wondering if you can tell me about the software you use. I definitely saw some R generated figures, but are there other figures that you or programs that you use to make your visualizations and or
Yeah.
slides?
Yeah, I'd be happy to share that. I do a lot of the sort of the plotting and the figure generation in R, as you said. I use ggplot too from R. Infernal is an open source software. And the other part of it is that the databases we use are public. They're completely open source. And so they're freely available to anyone who wants to do that. I will mention though, as a psychologist, not as I'm not a psychologist, but as sort of someone interested in sort of the translational aspect of this, it's long been known that there's, I think, an I don't recall the exact number, but I think there's like tenfold more serotonin in the lumen of the intestines than there is in the central nervous system. So thinking about how maybe the vagus is talking with the gut bacteria and maybe the way these bacteria are responding to our dietary and our host factors is really, really interesting.
Yeah, that is interesting. I did not know that about the serotonin fact. I feel schooled right now.
Just as it's an open invitation for collaboration. I'll put it to you that way.
Dr. Quarta, there's a question for you in the chat.
Yeah. Let me see if I could read. I'm just sorry I missed it during the case, but okay.
No, you didn't miss it. It just popped up. But
Oh,
the
okay.
person asking the question doesn't have the ability to unmute right now.
Oh
So
yeah,
she
no,
put it
no
in the chat.
problem. No problem. All right, so I'll just read it out loud. This was interesting. Thank you. I apologize that I can't unmute. No problem. You mentioned that your healthy subjects had higher than normal levels of more toxic chemicals such as manganese. And it looked like this was the case in the North American profile too. Yes, you're right. Compared to other parts of the world, it didn't meet statistical significance. So our conclusion was that there was no major differences. Did you look at the diets and dietary supplements of your healthy subjects? So we didn't have that available on this particular study. Or taking supplements that had things like manganese and the other toxic chemicals that could account for the higher levels in them in the gut. I think that's a great question. That's something, maybe supplements, or even, it doesn't even have to be necessarily taken in, but maybe it's something that's in our diet that's perhaps inadvertently being there, being introduced. Things, there's a concern, I think, among the general public that something in our diet could be actually harming us. Something, maybe the antibiotics that were traditionally given to livestock is actually making its way down into our diet too. And I will say that it doesn't just have to be in antibiotics. It could be other things. It's an open question. I don't want to, you know, make any strong conclusions. I don't think there's any data on either side of the coin, but I would say that perhaps those patients and those were healthy subjects. That study was actually out of India. So it was not in North America. I wonder if we were to repeat that study, if we were to see sort of different metabolic signatures in a different country. Uh, Let me keep reading though. Uh, okay. I wonder what we're consuming in North America that might account for those higher levels, especially supplements that we think are helping that are actually harming us. Yeah. I think, when patients ask me what vitamins should I take and what should I be doing, what probiotics should I be taking, I say I don't really recommend those things because we don't know what the companies are putting in them. I tend to shy away from things like that because I know I am biased and I'm sure that there are benefits, but I've seen a lot of patients with liver injury in the hospital from herbal supplements and dietary supplements. And yeah, I think probiotics are sort of also kind of an open question, an open area of investigation that we don't quite know what they're putting in. And so I like to use the example that, a healthy diet is a balanced diet, a diet that has fruits and veggies. And if you if you like to eat meat, you have a little bit of meat and chicken and fish and plenty of different things. And I think should be the major source of nutrition in our diet. Melatonin supplements with outrageously high levels compared to that what's advertised. Yeah, I came across a study about lead being contaminating applesauce and ending up in the applesauce of children in Australia, which is sad. But I think those are sort of one-off, very, very rare events. But I think it's something that we sort of might not fully appreciate. And it would be interesting to see if we sort of see different patterns among people. I still give my 6-year-old melatonin gummies. So do I.
Well, I just wanted to thank you so much for being our inaugural KL2 Visiting Scholar. So I'm Jen Krasniewski, and my question has actually shifted a little from your science to if you'd be willing to share what's been the highlight of your KL2 experience in your program. What can we learn from the great work that your team's doing there, and what could we possibly bring to Penn State?
Well, yeah, I think I think that the main thing for me was being selected and being given the opportunity to work on something that I'm very passionate about and that I'm hopeful will translate into improved understanding, science, and hopefully better outcomes for the general health of our population. And sort of a highlight, I think that you know, science, it's hard to say, that one moment, the eureka moment when, everything works and you discover something new. That takes time, and it's built up over a time, innumerable number of baby steps. So I don't think that there was any one moment except for being selected. That was like the one moment when I was like, yes, this is great. But I think that I'm just very appreciative and grateful. Okay, so what could be used? What's a model of where in some respects, we were really lucky and we were able to publish this. And I think that um, the model I used and the model I took was the following. Like, so I work in a basic science lab on RNA. It's really, really hard work. It's very challenging. It's highly technical, and it took me many years to develop sort of the gels and the mass spec and the transcription and all that stuff. But what didn't sort of take as long and sort of was sort of a nice little measure of something something achievable and something that I could do in the span of a year was sort of using, as Lauren mentioned, some of the computational tools that are out there on publicly available data sets. And sort of the basic science is there, and you can use some of the different tools that are out there, like R, like SAS, SPSS, to sort of look at the sequencing data in a very unique and interesting way. I think that's a model for a KL2 scholar that is we're all sort of under the gun, right? We have a couple of years and we need to hopefully produce something so that we can continue our work. And being able to do that in a timely manner, being able to get a data set that's already there and kind of ready for you is really, really valuable. So I think that worked in my example well.
Amazing. Well, I also just want to say thank you, Giulio, for joining us and sharing your science with us. And I know you're going to be connecting with some of our scientists here and people that are interested in your work. So if anyone has any last minute questions, I know it's 1 o'clock. Jess, do you have any closing announcements?
No, I think you handled it, LJ. Thank you.
Thank you.
Thanks, Giulio.
All right. All right, then I'll let you all go, and feel free to keep in touch if anyone's interested.